So, welcome back. Well, thanks for coming back. So this is the third part of my talk. And now I'm going to try and start explaining the problems that I alluded to in the first part. And before I forget, I just want to acknowledge the people in my lab. I'm not just going to talk about work from my lab, but I am going to talk about some of the work from my lab, particularly that of Thomas Glegoris, James Rhodes, and Naomi Patela, the key protagonists in some of the things I'm going to talk about. Now, so cohesin holds sister chromatids together. It also can build chromatids itself. But we discovered it as being required to hold sister chromatids together. We worked on the structure and the biochemistry of its core subunits SMC1, 3, and the cohesin, and discovered they formed a ring. But as you probably heard from Avram yesterday, that the transition from this state to this state is triggered by the anaphase promoting complex stroke cyclosome, APCC. What that does is activate separase, which is a huge proteolytic endopeptidase, and in fact the entire structure of separase has just been accepted for publication in Nature, not my lab, but that will appear, I believe, soon. It's a member of the CD clan of proteases, as we and Kunin discovered years ago. Now, what separase does, it's the activation of separase that causes sister chromatids to come apart at anaphase. And what it does is one very simple thing. It just clips that central domain of the cohesin subunit. And it doesn't matter where it cuts in there, as long as it cuts somewhere in there, that will cause cohesin to drop off chromosomes and sister chromatids will come flying apart. And so what this then led us to propose is that A, the discovery that cohesin forms a ring, B, that opening the ring destroys its connection with DNA, led us to propose the ring model in 2001. Namely, that the sister DNAs, this is drawn as 10 nanometer fibers, are trapped inside a single ring, and that would provide an explanation for how they're held together, and it would also provide a simple explanation for how separase triggers their disjunction. So that is the ring model, which we proposed over 15 years ago. Here is an electron, this is a very low resolution rotary shattering from Jan-Mikael Pedersen's lab of a cohesin ring before and after cleavage, in this case by TEV protease. But TEV can do the same thing as separase, as we've shown many times. You can hear these are the SMCs, this is the cohesin, this is the SSE3 subunit. And then and people say, oh, well, you can see rings, right? You see rings, but you don't know that's a ring. EM can't tell you that's a ring, but biochemistry can. If you put TEV in there and cleave it apart, and then it's do that, then you know it's a ring, you see. But anyway, okay. Now, so in order to probe, having got the structures of the various interfaces, there's these three interfaces that make up the tripartite ring, this one, and this one. And with those crystal structures that Jan Löwer had solved, with a little bit of help from us, we were able to devise cysteine pairs at the interfaces, which we could then cross-link together with bifunctional Thiol-specific cross-linkers. And we chose one, particularly this compound here, BMOE, which will cross-link them if they're sort of, say, 8-10 angstroms apart. So you need a crystal structure to get the cysteines in the right place. And the great thing about this is you can take a living cell and just put it on ice, add BMOE, and leave it for a minute, and then break open the cell. Basically, you can induce this cross-linking almost instantaneously in a living yeast cell. And then you end up producing this structure here. Now, using this cysteine-cysteine cross-linking, we can actually say, well, is cohesin really forming a ring inside living yeast cells? And we start off with a halo-tagged SMC3, and then if we have cysteine pairs here, here, and here. We call this 6C, not high C, 6C. Then what you can see is various intermediates, but basically we can get roughly 60% cross-linking at each interface, which means that you end up circularizing this thing chemically in about 20% of the molecules that this form here. Okay.
These are naturally occurring systems?
Huh?
These are naturally occurring systems?
No. We had to get the crystal structures and then put the cysteines in. The cysteines we had to show were functional, and we built a yeast strain in which you'd put in 6 cysteines. Actually, one of them actually turns out to be a natural cysteine, which is quite fortunate. But 5 of the 6 were unnatural cysteines. But this cysteine-cysteine cross-linking is actually, it's actually very powerful, rigorous technique for measuring interactions. And I believe, I think it's not been used as much as it should. Anyway, by this means, you can clearly show cohesin really does form a ring inside living yeast cells. Now, this then allowed us to test a very important prediction of the ring model. The ring model said is if you took a yeast cell with a very small circular mini chromosome, and basically you can have these very small, they're many about 2 or 3 kilobases in size, and if they were, if you built cystochromatid cohesion, they were held together by being trapped inside a single ring. And if we were using BMOE and a 6Z strain to chemically cross-link these 3 interfaces, we could then break open this cell, immunoprecipitate the cohesin, the DNAs would come down with the cohesin, the DNAs associated with this cohesin, we could then treat the cells with, we would then take the immunoprecipitate and essentially virtually boil it in SDS, but we do it at 65 degrees, and denature all the proteins. But if you chemically circularize the ring, these DNAs, even though they were, the topoisomerase had decatenated them, they would still be, as it were, catenated inside this circularized cohesin ring. And that was a very important prediction. So we did this experiment, and we had a control. We had a 6C strain, which is the experimental, and we had a control where we dropped out just 1 cysteine. So now you'd cross-link here and you'd cross-link here, but you wouldn't cross-link here, so you wouldn't circularize. And here is the result that you get. In the 5C strain, after BMOE treatment, all of the DNA associated with the cohesin and the immunoprecipitates is actually supercoiled, decatenated. So topoisomerase has decatenated the sisters. It's supercoiled monomeric DNAs that runs here in the gel. And then if you do the same experiment from a 6C strain, then you can see 2 new forms, and one of them turns out to be this monomeric DNA that's been catenated by a cohesin ring, and we call these catenated monomers, or CMs. And we know that these really are catenated because one possibility is you produce a little loop, and then topoisomerase could have knotted the DNA around the loop, and it actually, but we know that these DNAs are not knotted. They are non-knotted DNAs, so they really have been gone through that cohesin ring. We absolutely know that. And then the other form we're able to show is a pair of individual monomeric supercoiled DNAs that have been catenated by a single cohesin ring that runs up here. And we can show these, we can do 2D gels and have proteinase K in the second dimension, and so then these run as monomeric DNAs in the second dimension if you do proteolysis in between the 2. Huh? Oh, that's a good question. I don't want to go into that. We've done experiments where we've made diploids and we've got 2 different versions. It's a complicated. I don't have time to go into that, but we have done reasonably rigorous experiments to show that it is a single monomeric ring. Okay, so
you know from its separation?
Absolutely, yes, you could. Yes, so these would be destroyed. We've never done that with separase because it's difficult to make, but it's certainly yes.
Do the cohesins bind proteins somehow? How can they?
The cohesin doesn't really bind chromatin. If you take if you take Roger's favourite polytene chromosomes and you have a Drosophila which has got TEV sites in the Kleisen subunit and inject into the salivary gland TEV protease, then all the cohesin on the polytene chromosomes comes off within a few seconds. Okay, it does not bind chromatin. I mean, I'm sure it does bind chromatin, but not very tightly. Okay, so essentially, according to the ring model, the association is topological, and all the evidence we have suggests it's topological. Okay. Now, so we think that these then is the product of DNA replication. CDs, we suppose, are synonymous with cohesion, and CMs would be synonymous with cohesin going on to chromosomes before replication. So if that's the case, if we were to take a yeast cell in which in the wild type it was allowed to replicate and it produces CM and CD, but if you prevent it from undergoing replication by blocking degradation of the yeast equivalent of P27 that Avram talked about yesterday, then the cohesin's made, it goes onto chromosomes, they don't replicate, you don't form CD, but you still form CM. Okay, so that and a whole series of other experiments looking at various different mutants that can or can't bind to chromosomes, that can or can't build cohesion, we have so far found a perfect correlation between whether or not cohesin can load onto chromosomes and whether or not you form CM in this assay, and a perfect correlation with whether or not you can form cohesion and whether or not you form CD in this assay. So what we believe, therefore, is when cohesin loads onto chromosomes, it traps a DNA, and that's loading, and we can measure that with these small mini chromosomes as CMs, and when you undergo replication and hold sisters together, then you're trapping the two DNAs inside these rings. That's what we believe we can conclude from these experiments at the moment. And this gives you an idea of the scale of the beast. I think this is quite a useful one. This would be a 10 nanometer fiber, and this I've made up the coil here, but this is a real structure, this is a real structure, this is a real structure, this is of course a real structure, and bits of this are real and bits that I've just made up. But this is the size of the Kleisen, okay, and this is the bit that binds those hawks, okay. But that gives you the scale of the beast. It's big. There's lots of room for diffusion inside there. And that would be the CD. And then again yes. So that's what we think. Loading is entrapment of DNA inside a ring. Cohesion is co-entrapment of sister DNAs. That's our working hypothesis. Now, I've told you about...
so the replication fits in with the size?
In principle, yeah. In principle, I'll come to that. I mean, I don't have the answer, but I'll come to something that's relevant. So the first thing I want to consider is how, just forget about replication for a moment. There's just this reaction. Let's take a cell that's not replicating. Cohesin goes onto chromosomes. And I told you need this SSE24 loading complex. And then, in fact, in all of us, in us and in yeast, cohesin can go onto chromosomes, and then it will also come back off. So it's actually turning over on chromosomes. In a yeast cell, it's turning over the half-life of a few minutes. In you and me, it's probably 10, 15 minutes it's turning over. So it's basically loading and releasing, loading, releasing. And loading requires this SSE2, SSE4 heat, this hawk, and releasing requires the PDS5 together with WOPL. And it also requires this SSE3 subunit, and loading appears to require this SSE3 subunit as well. Okay, so you need one set of hawks to get on and another set of hawks plus WOPL to get off. And according to the ring model, loading, somehow you've got to get the DNAs in, and release, you've got to get the DNAs out. That's what we think, according to the ring model. Yes, I shouldn't have done that, should I? Yes. Okay, now, we don't know a great deal about these reactions, but one thing we are most certain about is so the big question is, where are the entry and exit gates for DNA? And we don't, what we know most about is the exit gate, namely release involves the dissociation of this N-terminal domain of the Kleisen from the coil associated with the SMC3 ATPase head. This is really quite good evidence. Basically, if you fuse this bit of protein to this bit of protein here, cohesin goes onto chromosomes, can't come back off. In addition, what we know is that this releasing reaction is taking, so if this goes inside, then releasing activity will get you back to here. And in principle, you can build this structure and releasing activity would destroy that as well. So, how do you build cohesion and keep it stable? Well, what happens is that the generation of these structures at DNA replication is accompanied by the acetylation of SMC3 by this acetyltransferase called ECHO1. And during replication, at least in yeast, it gets acetylated, and what that does is it shuts off this releasing activity, so that once you're acetylated, then you can no longer be released. Okay, so the releasing activity is shut off by acetylation. And you and me, you need acetylation plus another protein called serorine, but don't worry about serorine. Yeast doesn't have it, so it's not important. Now, what sequences are acetylated? These are a pair of lysines, very conserved lysines here. In the ATPase head of SMC3, they're pretty close to the interface, which is opened up by releasing activity. Somehow their acetylation prevents this being dissociated from the coil here. And we've been able to show, we can actually measure this release, again using this BMO-induced crosslinking. So it turns out the separase cleaves the N-terminal fragment, and after separase cleavage, this will remain associated with the SMC3 if you inactivate releasing activity. And you can measure that by the cysteine-specific crosslinking. But in fact, if you have releasing activity, this is destroyed, and you as it were, don't get this fragment is no longer cross-linked to the SMC3. Well, what I've actually shown you here is a wild-type cell where releasing activity has destroyed this. This has been dissociated, and here is a QQ mutation here. And basically what this shows is an acetyl mimic QQ, as it were, blocks the releasing activity of this fragment from the SMC3. So this is not, Avram wouldn't approve of this. This is not biochemistry, but it's what I call in vivo biochemistry, which I think is extremely important to be done that precedes the real biochemistry. And Frank Ullman has got some data suggesting you can get this to work in vitro as well. Okay, so that's release. Very important reaction. The mechanism of loading is less clear. According to the ring model, you've got to open up the ring and get the DNA inside. And we have proposed, on the basis of a number of experiments, but it's certainly not proven, is that the entry gate happens to be this interface up here. But this remains extremely controversial, and I don't want to go into that. But our hypothesis at the moment is that when the heads engage, somehow this then leads to the dissociation of this interface, creating a gate for the DNA to come in. And then when you hydrolyze, they shut again, and the DNA is trapped inside. That's our working hypothesis. Don't accept that. We don't know if it's true, but it gives you a way of thinking about it, okay? But this is one of the big questions in the field. Now, somebody asked, what happens at replication? How do you build these structures? Could you drive the replication fork through a cohesin ring that's already there? Well, I can't give you an answer to that question, but what James Rhodes in my lab has set out to ask is, what is the fate of a cohesin ring on an unreplicated chromatid after a replication fork has gone through? Does it remain there after the fork has gone through? Now, we haven't been able to do this at a single molecule level, but we've done it at a level at which I think we've got the answer. And what James has done, we've moved from yeast now to mammalian cells because the imaging is superior. He's taken U2OS cells, and to do this experiment, we needed to, and what he's done is he's developed, and this is a very, very important technique that I think he's developed, he's developed a way of labeling proteins and pulse labeling them using the halo tag. People have started to use this for measuring proteolysis, but nobody's actually done this in the microscope. And a lot of these FRAP experiments everybody does are all fraught with the fact that the fluorescence always recovers because you're always synthesizing new protein. So what James does is he takes U2OS cells and halo tags the cohesin subunit SEC1, and it's homozygote. You know, we know it's functional because all the copies are tagged using CRISPR. And then you take these cells and treat them with a fluorescent ligand that will then be attached to the halo tag, the halo domain, and you label up the cohesin using this dye. And then you wash away the dye, and then you come in with the ligand without the dye. And so now you prevent, if there is any dye lingering around in the cell, which there will be, the unfluorescent dye will prevent it ever labeling any more protein. So the only protein that's been labeled is the stuff that was labeled during the pulse. It's just like a pulse label experiment. And so that's allowed us to, so this is what happens if you don't do this this chase with the dye without the ligand. You can see that within 16 hours, if you completely photo bleach this stuff, you can see it recovers, okay? But if he chases with the ligand, no recovery at all, okay? So this is a really very, very simple technique, but it's a very powerful one. So he then took these cells, and he started off with either wild-type cells, and then he took, again using CRISPR, he knocked out the WOPL subunit. And WOPL, as I told you, is required for release, but it's not required for loading. And so now you prevent any cohesin turnover. It goes on the chromosomes, never comes off. Now, what you can see is in wild-type, the chromatin is a smeary mess all over the place, and you can start to see these axes of the chromosomes, just even in these cells, in living cells here, when you've knocked out WOPL. Okay, so we're looking at cohesin, the halo tag cohesin. Now, what happens in wild type is the releasing activity goes into hyperdrive when cells go into mitosis, and only the cohesin at the centromeres is retained during mitosis. And you see almost all the cohesins come off into the cytoplasm. You'll just see the centromeric cohesin. But when you knock out WOPL, then none of the cohesin comes off chromosomes, and cohesin you see all the way along the chromosome arms throughout mitosis. But these cells go on proliferating. Just you knock out WOPL in yeast, they go on proliferating. And in fact, yeast doesn't have this dissociation pathway. Basically, you turn this mammalian cell into a yeast cell. And all of this stuff has to be cleaved by separase. Okay?
Scenario of WOPL knockout, is condensing still essential?
Good question. Almost certainly, because remember, condensin is not cleaved by separase. But you absolutely hit the nail on the head. I think that you could possibly get rid of condensin if you knocked out WOPL, but the trouble is it's not regulated. Cohesin would get cleaved by separase, and so then that's the problem. But if it weren't for that, I think the answer is yes. This guy is on the button. Okay, now, so here's the experiment. We've got these cells. And here we've in fact got the halo-tagged SEC1, and we've got a snap-tagged histone H3. And we do this pulse chase experiment with two different dyes, and what we do is we bleach all of this and all of this. So we basically end up with two little strips. We either label the same strip is either labeled with the snap tag for H3 or for the cohesin, okay? And then we chase it for about a cell cycle, and what you can see is the histone H3 stays more or less in situ. Some of the chromatin wanders. You see it's wandered down here, but most of it stays in the same place in the nucleus after 24 hours. But the key point is the cohesin is in exactly the same places, okay? So everyone's known for a long time H3 doesn't turn over, and at least in a WOPL mutant, cohesin is not turning over, and of course in this experiment it's co-segregating with the H3. Now here's the experiment. What happens to this labeled cohesin when a cell undergoes replication? So now we label up pCNA using GFP, and we can, this is living cells, G1, they go into S phase, you see these replication factories, and they finish S phase into G2. You labeled up a certain fraction of the nucleus, you chase it through S phase, it turns out the cells are moving, and this crescent here turns into this crescent here, but it's the same part of the chromatin. And what this tells us is that this cohesin that was on the chromosomes on this particular patch of chromatin before S phase ends up on the same patch of chromatin after S phase. Okay? So somehow the cohesin is not displaced by the replication fork. Now, whether that means that the fork can go through, basically what we think this is telling us is that this cohesin here, as long as you prevent turnover via WOPL, of course if it's turning over you wouldn't be able to do the experiment, but we block that turning over, but it's not displaced by the fork, and we think this turns into this. Now, how it gets from here to here, I can't tell you. Does it go through, does the fork go through it, or does it somehow open up but not let go? I don't know. This single molecule experiments will, that's going to take a little while. It's a very interesting question. But at least we've defined the nature of the problem. So that's James's, a large fraction of James's PhD thesis. But it's a very nice and very simple experiment. Now, I want to move on to something that should be simpler, is the loading reaction. Let's forget about what's happening in replication. So how does cohesin get onto chromosomes? What's actually going on? And people have started to do a lot of this with in vitro systems, but there are some problems with the in vitro systems. This harks back to what Avram was saying, is the key thing about in vitro system, it must replicate what you know what's going on in vivo. And we know in vivo that loading requires ATP, it's knocked out completely by EQ mutations, it's completely dependent on SEC2, it's certainly not dependent on WOPL or PDS5, and yet a lot of the in vitro systems where people can measure cohesin loading, either in bulk or in single molecule experiments, are either independent of ATP or independent of SEC2 or driven by PDS5 or WOPL, which for me, as a biologist, rings a lot of alarm bells. I do believe we need to go down the in vitro system, but I think one needs to be very careful what one concludes of systems that are not showing the right properties. So I think there's therefore a need to measure more accurately what's actually happening at loading sites in living cells, and then you would have a better idea of what it is you should be trying to reproduce in vitro. Yes?
Are those loading sites determined? Or
I'm kind of come to that. The answer is we know very little about loading sites in any system except for one, and that's in the budding yeast, Saccharomyces cerevisiae. I'm going to talk about that now. This is the work of Naomi Patela, who's just graduated from my lab. Now, what Naomi did was to develop, with Bin Hu in the lab, people have been doing ChIP-seq for a long time, but ChIP-seq gives you a distribution. But what you want to do is to be able to compare different samples. You want to say, is there more or less protein at this position in the genome between sample A, sample B, sample C, sample D? ChIP-seq cannot tell you that. But we developed a system. There were a couple other labs that did it at the same time, but I think we did it somewhat more rigorously and in a way that people can understand what we did. It's a little paper in NAR, but it's one of the best papers I published in the last 5 years. So we call it calibrated ChIP-seq, and we basically mix different yeasts together. But basically we can take these ChIP-seq distributions and calibrate them so that we can actually measure the level of cohesin genome-wide in different samples. And here's an example of the distribution of cohesin along an entire chromosome as cells go through the cell cycle. They start off in G1, they replicate, they undergo anaphase, separase, cleaves it off the chromosomes, they re-replicate, cohesin goes back on. So not much cohesin in G1, goes on just before S phase, stays on until anaphase, separase, cleaves it off, you remake it, goes back on again. Now if you just did conventional ChIP-seq, the area under the curves of all of these would be the same, by definition. It's completely worthless. And I will tell you, 100% of all the ChIP-seq experiments that have ever been done are 90% worthless. And they're completely misinterpreted by everybody who's ever done them. Okay? For that very simple reason. The area under the curve is the same in all samples. You have to calibrate them in order to be able to get this sort of data. Okay, now, so we can take this assay and put it through the motions. Well, I've told you need this SEC2 protein to get onto chromosomes. Calibrated ChIP-seq if you send cells through S phase with SSE2 active, this is what you get. And in a TS mutant at the high temperature, this is what you get, okay? Very nice, and that correlates with our CM-CD assay, okay? At the high temperature in this mutant, you don't get any DNA co-precipitating with cohesin, you get no CM, no CD, okay? These two guys track together, okay? These two assays. Very different sorts of assays, but they go together. Now, loading sites. Everybody knows this is how our chromatin is packaged inside the cell, except at this very specific place, which is at kineticals, where you have this centromere-specific histone SEMP-A nucleosomes. Now, in most organisms, you have multiple SEMP-A nucleosomes and multiple microtubules. But about 50 million years ago, in a narrow lineage that led to the budding yeast Saccharomyces cerevisiae, and most of what is good about making wine and bread and whatever it is. But anyway, there's in the Saccharomycete, something dramatic happened, and you got rid of all of those SEMP-A nucleosomes except for one, and you only just a single microtubule attaches via this wonderful structure that Harrison's lab has just produced to that nucleosome. Okay, so essentially, the yeast centromere, as shown many years ago by John Cartman, is defined by a 100 base pair sequence. A site-specific DNA binding protein binds to a site, brings in this nucleosome, microtubules attached to all those wonderful kinetical proteins. But you have this incredibly simple structure. Now, the point is the following. Centromeres are where a lot of the pulling is going to be done, and that's where you need most of the cohesion. Okay? And in a normal centromere, where you've got lots of SEMP-A nucleosomes, you've got lots of sites to load lots of cohesin. But now you produce this streamlined centromere, and now this particular nucleosome has got to work much harder to load enough cohesin to resist the force of the microtubules. And so it turns out that these centromeres, these point centromeres in yeast, are turbocharged loading sites for cohesin. Okay? And this was really first worked out by Paul McGee's lab. He was in Denver. I'm not sure what he's doing now. Here's an example of this. It turns out that if you take all 16 chromosomes and align them on this point centromere, and then plot the average of the cohesin along the chromosome out along the chromosome arms. This is just going 60 kilobases on either side. If you have a wild-type centromere, then what you find is in about a 30 kilobases on either side, you have very high levels of so-called pericentric cohesin. But if you have a centromere that's defective in this centromere-specific loading, then you see it's just like any other sequence along the genome.
What's the spacing to the first peak?
What is the
spacing?
That will be. They are about 300 base pairs away from the centromere. We can go in, it's interesting. Yeah. And this is the experiment. This is an experiment we did, but it's a repeat of the experiment McGee first did. If you take a particular chromosome and remove the centromere, so normally here's the centromere of chromosome 14, and this is this pericentric cohesin on that chromosome. And then if you remove just that point centromere, you see all of that heightened levels of pericentric cohesin disappears. And then you put it in an ectopic site where normally there's not a lot of cohesin, and now in the surrounding region you now get very high levels. So that point centromere is both necessary and sufficient to drive the association with pericentric sequences of around 60 kilobases in interval of very high levels of cohesin. The idea essentially is that the cohesin is loading at the centromere and then translocating down into the pericentric sequences. And we think we can see this. This is again reanalyzing that synchronous culture using these average chromosomes. And this is the G1, this is 15 minutes after release from the G1 arrest. And then suddenly you make this burst of cohesin. You can see it's starting to arise, this red curve. You've suddenly gone up, high levels of cohesin have loaded between this time point and the red one. And you can see it's preferentially associated around the loading site, and then with time you can sort of see it spreading. It's not a pulse chase experiment, it's a poor man's pulse chase experiment. It's the best we can do at the moment. So what we think is going on in yeast, and I hope this answers your question, we believe the centromere is a specialized turbocharged loading site, and cohesin loads on there, traps the DNA, and then it can translocate up to 30 kilobases, possibly even further down the chromosome. Okay? Now, so do you see, we think cohesin's coming on at this site. You might imagine you would also see the loading complex at that site. And lo and behold, you can see, it turns out both in G1 and in cycling cells, if you look at the distribution of the loading complex, the levels of association are very low compared to cohesin, but you can see this huge peak right at the centromere itself, okay? Right at the centromere, you see high levels of loading complex. You don't see much anywhere else on the chromosome. So, interestingly, one of the questions you can ask is when you see, we know that cohesin arriving at this site depends on the loading complex. Does the loading complex being present at this site depend on cohesin, the reciprocal? And Adele Marson has published some experiments showing this was the case, but we've gone back and revisited that using this calibrated chipset. And what she claimed is indeed correct, that the arrival of the loading complex with time as cells goes through S phase in the wild type does not take place if you allow the cells to enter S phase in the absence of the loading in the absence of cohesin. So basically, cohesin coming in here depends on the loading complex, and the loading complex coming here depends on cohesin. They're interdependent. And it turns out, so then there's sort of two possibilities. Does SSC2 form a complex with heterotrimeric rings during loading at sense? That would be one possibility. During the act of loading, the loading complex forms a complex with cohesin. And that's a transient intermediate of the ring, and then it traps and moves off, and the loading complex lets go. Or is the loading complex merely recruited to centromeres by kinetochore proteins like the CTF19 complex, where it catalyzes a change in the behavior of these rings? So here I think we've been able to answer this question. This is Naomi's work, and we go back to this whole idea that loading depends on this ATPase cycle. So you need ATP to bind, heads to be engaged, and then you need hydrolysis. And there are some very specific mutations, either this mutation in the SMC1 ATPase or the equivalent mutation in the SMC3, which allow the ATP can bind in both cases, but you can't hydrolyze either of the ATPs. And so we think that the heads can be engaged and then they can't be disengaged, okay, in these single mutants of either SMC1 or SMC, these EQ mutations. Now these are lethal, these mutations, because you can't load cohesin onto chromosomes. And this is the, again, calibrated chipsec of wild type. I put it on a completely different scale. This is wild type cohesin, 15 kilobases either side of the centromere. And this is what you see with an EQ version of SMC3. You just see it right at the loading site, okay? And very high levels. On a different scale, this is the wild type. Very little EQ on the arms, but actually at the loading, this peak going right up here. So these EQ guys, if you prevent the hydrolysis of ATP, the cohesin gets the loading site, and then it can't translocate away. And it turns out you can see these complexes accumulating in live cells, and you can see that it's actually going through, it's turning over with a half-life of about 5 or 10 seconds. So it's going on and dropping off, going on, dropping off, going on, dropping off. It can't get into the pericentric sequences. And we know that it can't trap. You can't form either CM or CD with these EQ cohesin. So that's what we think is going on. These EQ complexes, they form this intermediate at the site at centromeres, but then they drop back off again. No entrapment, no translocation. Okay? Now we can now take a wild type cell. We've got wild type cohesin, which we believe is loading here and translocated into pericentric sequences. And then we express in the cell some of this EQ SMC3. So we have two populations of cohesin, wild type, which can load and translocate, and EQ, which can only go to the loading site and then will just drop off again. So we now do calibrated ChIP-seq in these cells, and we're going to see two mixed populations, wild type plus the EQ. Okay? Now, if we look at the cohesin subunit, in a wild type alone is the blue curve. Okay, basically it loads here, translocates. You don't see much at the loading site. But if you express EQ, now we're seeing the second population on top of this other population. You see that this stuff is still there, but now you see a new population appearing at the loading site. That's the EQ cohesin at the loading site. Okay? Well, this is lovely. We can now play this game and ask, what subunits of cohesin are present along with the EQ cohesin at loading sites? Well, and basically the SSE3 subunit, we can see it there. It may be a little bit less than we would have expected, but it is there. And crucially, what you see is that the SSE2 subunit of the loading complex, this is what you see in wild type. Those were those pictures I showed you before. And now you express EQ, and now what happens? Boom. You go from here to here. So essentially, SSE2 is part of this EQ cohesin that goes to the centromere and then drops back off again. It's actually part of that cohesin complex at the loading site. Because just preventing it from completing hydrolysis causes it to accumulate to massively high levels. Now, I told you there's three Hawk proteins, SSE2, the loading complex, SSE3, and PDS5, all structurally homologous. What about PDS5? Look what you see with PDS5. The red line is the EQ guys, the blue line is the wild type, and you see there is no PDS5. PDS5 is not increased at the loading site by expressing EQ. Okay? So what that tells us, I believe, is the following. Is that in the nucleoplasm, the cohesin rings are associated with PDS5 and SSE3, these two Hawk proteins. And the heads engage, and what happens is that the SSE2 Hawk displaces the PDS5 Hawk, and we believe this opens the ring up. So when the heads are engaged, which will what will happen when you have an EQ mutation, SSE2 is bound instead of PDS5. You open up the ring and you attempt to do a loading reaction, but then you can't hydrolyze and you drop back off again. But what would normally happen in wild type is you get to this state and then you hydrolyze ATP, you shut the ring, you entrap, and now the SEC2 is displaced by the PDS5, and now the version of cohesin you see on the chromosome is the version that contains PDS5. So basically, SEC2 is not really, it is really a cohesin subunit. It's just one that's only there when you engage heads, in particular happening at the loading reaction. Now, it will become clear later on, and this is a very important issue, we know that the cohesin complexes that have translocated and that are stably associated with chromatin and which you can measure by ChIP-seq, they contain PDS5 but not SSE2. I've told you it's SSE2 that drives ATP hydrolysis. I've told you that these guys can translocate on chromosomes. Do you need ATP hydrolysis? Do you need SSE2 for these guys to translocate down chromosomes? That is, what may be happening here at the loading site may also be happening, albeit very fleetingly, during the translocation process. We do not know. But is that clear?
And what prevents them from falling from the chromosome?
Good question. I don't have any answer. We do know that normally WOPL would open up this thing and it will drop off after a few minutes. But the question is, if SEC2 is transiently displacing PDS5 during translocation, what would prevent it? Absolutely. But if you want my guess, these guys, it looks like they're going for megabases in you and me. I do not believe that's ATP independent. and I believe it's going to be driven by SSE2 or NIPBL. I think SSE2 is playing a fundamental role not only in the loading reaction but also in translocation. But that's a guess, a total guess.
So you don't believe this idea that transcription drives these?
No.
these movements?
Transcription will influence where you end up. It will influence the steady state. But these guys are going, if you look at the maps, first, no, the answer is no. In bacteria, where we think the same thing's going on, it's going all the way down a chromosome arm. In yeast, they're going against the grain of a lot of transcription units. If these guys are going 30 kilobases or even further, they're going through a whole series of transcription units that are facing the wrong way. But it is the case that all things being equal, they will tend to end up at the 3' ends of genes. But that is not the same as saying it's transcription that's driving it. I do not believe it's transcription that's driving it. In fact, I know it's not. Yeah.
How many complexes like this actually can?
Well, there's a whole business of traffic, and I lose sleep over traffic. Okay. Literally, I don't know. It is a problem. It's a problem. But the key thing about hypotheses: don't expect them to explain everything immediately, because if you try and explain everything, the hypothesis inevitably is ugly and undoubtedly wrong, because most of the things you're trying to explain are themselves wrong. So don't try to explain everything. I don't know if traffic's a problem, so yet.
Could microtubule forces be part of what's driving them?
No, it's nothing to do with microtubules.
So what are these valleys?
Huh?
What are these valleys between the paracentric?
Oh, I don't have an answer to that.
It's not repeated sequence.
Huh?
It's not just intrinsic in the bioinformatics. It's not because it's repeated sequence.
We've looked to try and see what it is, and I haven't been able to work it out, but I haven't got anybody to work on it terribly carefully. Yeah.
So the cohesion ring is loaded at the centromere.
Well, it's actually, cohesin is loaded throughout the genome. I believe it's loaded throughout transcription units in yeast and you and me. But over and above that, you've got this very special loading mechanism at centromeres in yeast.
My question is different. I mean, you have the cohesion ring and you have the replication fork, and
But this is nothing to do with replication.
I know, but I
Yeah.
mean, the replication doesn't happen when you have cohesion on the.
Oh yes, what happens to these things during replication?
Something else. You have the replication. You have recombination-based events.
Yes.
How do you prevent the cohesion ring, the sliding of the cohesion ring, from being stopped or disrupting these recombination-based events?
I have no idea.
Okay.
These are all, I have no idea. Absolutely, I have no idea. Okay. So what we believe is that SSE2 displaces PDS5. Now we happen to know, Jan and my lab were able to get a structure of PDS5 bound to the cohesin, and we know that the place on the cohesin that PDS5 binds is this region here. And Hung-Tao Yu's lab has just published a structure of SSE2 from a fungus, a chetomium, but it'll be not very similar to yeast. And he has found that SSE2 binds tightly to an equivalent region, a similar region of the cohesin. And so what we think is going on is that that PDS5 and SSE2 are competing for the same region of the cohesin, and SSE2 can somehow kick PDS5 out when the ATPases are engaged, and then PDS5 can kick out SSE2 when they disengage. Okay, so these two subunits are the yin and the yang of cohesin. So a key point of this would be that the region where we think SSE2 is coming in and binding should be necessary for loading. And here is a deletion that we've made recently. Here's a wild type where you get the loading here, and here's this deletion. It forms perfectly nice protein, and you can see that it can't load onto chromosomes. So we believe that our work, together with Hung-Tao's work, has led to this idea of SSE2 and SSE2 having to displace PDS5 from the cohesin transiently during the loading reaction, and possibly also during translocation. This is PDS5 bound to a little fragment of that cohesin. This is a structure from Jan's lab that we published last year. This is the structure, or Hunk-Tau structure of SSE2, and you can see how structurally homologous they are in this region. This is the PDS5 and the SSE2 together, and you can see in the region where we know the cohesin's binding to PDS5, the SSE2 and PDS5 are structurally very similar. So you could see these two guys competing for binding to this region. So to summarize this part, I want to just say we think that in the nucleoplasm you've got these rings, you've got the Hawks, PDS5 and SSE3 bound to the cohesin. At the loading site, when heads get engaged, SSE2 displaces PDS5, you open up the ring, DNA gets in, you then hydrolyze the DNA, shut the ring again, and then PDS5 comes in and displaces SSE2, and then you translocate down the chromosomes. So that then is what we think is going on in yeast. And this has taken us nearly 20 years to get to this position. It was nearly 20 years ago that we identified the first mutations in the SSE1, the SMC1, the SMC3, and the SSE2. And this was not biochemistry, this was genetics. Okay, now, more problems. Okay. A criticism of the ring model. Yes. Okay, well, maybe I could stop here, or I could talk about loop extrusion. I don't know what you want me to do.
How much?
I probably need 10 minutes.
I want to hear everything, so
Okay, very quickly. Here's a major criticism that actually Tim Mitchison made. It's an acute one. He said, condensin and cohesin are related complex. There's no evidence that condensin functions by entrapping DNAs. Therefore, this principle cannot apply to cohesin. I fully understand the logic. However, I came to a different conclusion. If we assume that cohesin works as a topological device that entraps DNAs and that cohesin and condensin use similar principles, then how could condensin operate as a DNA entrapment device and organize DNA into chromatids? You see, Tim and I started with the same place, that they got the same activity. He concluded I must be wrong. I concluded that he was wrong about condensin. And that really was the basis of proposing the loop extrusion model. When we realized that cohesin might work as a topological device as a ring, I said, how might condensin organize chromatids using a similar principle? And that led me to propose the loop extrusion hypothesis. The idea was somehow condensin and also cohesin can entrap somehow little loops of DNA. It could either be a single ring or two rings, we'll see later, we have no idea. But the key point is once it made these little bites in the DNA, that then somehow the DNAs could translocate through these rings and the loops would get bigger and bigger, but it would be done in a topologically clean way. Okay, so the DNAs would be passing through the rings, and so any sequence that emerged in these loops would be cleansed from the rest of the genome. And we just said that, condensin was a dumb enzyme. This guy didn't know what this guy was doing. They all did it together, and so they kept on doing it until they packaged the chromosome up into a cylindrical structure with loops emanating away from an axial core in which you had condensin running along the axis. And I knew that cohesin was doing the same thing in meiosis, so I said, well, cohesin must also have this property. Nothing happened for 12 years, and then John Marco came up with the same idea, I think independently. He didn't notice my paper. And then there's some beautiful simulations have been done by the Mirny and Marco labs. I just want to show you a very quick version of this. Yeah, this is one of Mirny's simulations. I don't think these simulations prove a great deal, but I think they just shows that, I did it with ropes and carabiners, but they've done it with computer simulations, and this would be a single chromatid with red at one end and blue at the other, and these are very nice ways of illustrating this principle and help people to think very clearly about it. And the same, of course, cohesin would do the same thing as well as cohesion. Now, the big I think the big explosion in the field took place last year when the Lieberman and Aden and the Mirny labs suggested that loop extrusion could explain key features of the high C patterns that everybody's been seeing, and I alluded to in my first talk. And here is that fundamental pattern, the TAD. What are the key question that emerges that you'd like to know the answer is, what is a TAD? The second question, why are there particularly high frequency interactions at the borders of TADs? Okay, it's the question number 2. Question number 3, why do high frequency interaction sites at TAD borders co-localize with cohesin and CTCF? Question number 4, and this is the real humdinger, is CTCF sites are asymmetric. Why do sites at TAD borders usually, in 90% of the cases, face each other? And this is a very, very crucial issue because these can be megabases apart. How do they know which way they're facing? Here is the problem. You've got, and then somehow these guys know which way they're facing. These will associate with each other at high frequency, but not if this is pointing the other way. And this is an absolute, and you expect this to be in 25% of the cases, you actually see it in 90% of the cases. And this is really one of the most interesting things that has emerged from sequencing the human genome, in my view, apart from all those genes. But this is a real mystery. Okay, so what is responsible for high frequency interaction between TAD borders? Well, presumably somehow these TAD borders are brought together, cohesin is present there and is somehow holding them together in a loop. And one possibility is, of course, that they've been trapped inside a cohesin ring. And then the question is, what is a TAD? And one of the ways that people have thought about it is you form this loop and you form a neighboring loop, and then if you're part of this loop, you're more likely to be cross-linked in this ligation reaction than if you are here. Well, this has got to be wrong, because it turns out, if you look carefully at these guys are actually very infrequent. So these structures are very infrequent. They look frequent, but they're actually very infrequent. And therefore, there's no way you can explain this hugely high frequency within the whole TAD by this rather infrequent sites here. That cannot be the explanation for what a TAD is. But that's what you'll see in most of the reviews. So the other possibility, I think, and I think Mearney thinks probably the same, is that if a TAD is merely an interval within the genome within which most combination of DNA sequences throughout that interval can be brought together by being extruded through the same cohesin ring. So you initiate this loop extrusion process at random in an interval, and you initiate here or here or here, and then it goes to completion, and then the extrusion stops when you get to CTCF sites that are facing in particular directions. Now, what creates the TAD is not generating this structure. It is the process of this sequence is being brought together by this through the process of being extruded through the same device. And so if you take an amalgam of all these events throughout this interval, and if you assume it's random, you would generate the TAD. And I believe that is what a TAD is. And so the question is, how would cohesin create these small loops, and how would cohesin extrude such loops? Now, I've told you that in yeast we believe that the loading reaction looks like this, and then you translocate down the chromosome. And the big question is this reaction here catalyzed by SCC2 and ATP? Well, what if you have two rings that started translocating down the chromosome and bumped into each other, and at that point they then, when they then hit each other and something happens, they hold on to each other, but then they start translocating in the opposite direction, then they would end up extruding a loop through a pair of rings like this. This is just a hypothesis, okay? But it's the hypothesis builds on our current knowledge of cohesin, but don't take it too seriously. And so if this occurred throughout the genome, this is occurring here, here, and here, and all the way through this interval, and you end up creating the structure here. Now, so according to the above version of loop extrusion, TADs are not real. They're not real things. They're merely a property of the genome that emerges from the process of loop extrusion, and a process that is punctuated by CTCF. And now, of course, that's what everybody refers to as TADs, is these boxes here and these TAD boundaries. But actually, if you look at Erez's data, at least half of the TADs don't look like this. In everybody, there are lines all over the place. And what are they? And I think what those are is that this process of extrusion might not be completely random, and that there are actually, these are actually, we were looking at the extrusion tracts. But that's just a hypothesis.
The border is
It's highly asymmetric.
so therefore
It's almost as if you're starting at one end.
sliding. It's like one thing is
Yeah, yeah. But sometimes you see it, yeah. But anyway, these high C things are full of these lines, these tracts.
Did they perform high C CDF mutants?
It has been done. It's not published, and yes, I haven't seen the data. I've just heard about it,
So
and yeah.
what is proposed to be the force for the extrusion? I mean, you need an ATPase or some strong.
Yeah, and that's why I talked about this translocation. Do you need the ATPase for this translocation? And
This is different from translocation. This is like a pumping. I mean, you're pumping huge amount of DNA.
well, I guess the force is thermal motion, and because this is not a very strong ATPase, and the ATPase is to sort of regulate the directionality. I don't believe that it's not a powerful enough ATPase to act as a motor. I think the motor is thermal motion, and this is regulating thermal motion. But that's, what do I know? I'm not even a biochemist, so. And could cohesin rings that hold sister DNAs also extrude loops? Okay, then what's the relation between loop extrusion and cohesion? Well, you can see they're not necessarily incompatible. So many questions. You've got most of these. And I just want to say that now the bacterial people also think that loop extrusion may be the mechanism by which the two arms of bacterial chromosomes are segregated each other. So in B. subtilis, the work from Rudner's lab, and is basically it's thought that the bacterial complex is load on close to the origin at the par S sites, and then you extrude the loops, and then basically these guys run down the chromosome following the replication fork until they get to the terminus. And as a consequence, you end up zipping together the two arms of each sisters and thereby segregating them away from each other. Now, I don't need to say anything about that except that, say, cohesion is very important in cancer. And I just want to end up with this sort of I think these lovely quotes that I think are sort of different ways of saying the same thing. Loop extrusion was a complete fantasy I had after coming back from a climbing trip and fiddling with a lot of climbing ropes and carabiners. But what I was trying to do is say, I knew you had to explain X, and I postulated a piece of sort of invisible simplicity to explain it. And this is a quote from Jean Perrin, and I think it's appropriate here to say this in an institute that's dedicated to Einstein, because Perrin is the guy that took Einstein's idea that atoms could explain, the invisible entities atoms could explain Brownian motion. It was Perrin who proved Einstein was right. And at the beginning of his great book, Atome, he has this thing: we certainly observe these visible pieces as closely as we can, but at the same time we seek to divide the hidden gears and parts that explain its apparent motions. Our task is to explain the complications of the visible in terms of invisible simplicity. Loop extrusion is simple, it's invisible, and as Jacob puts it, slightly differently, but he's saying the same thing. The process of experimental science does not consist in explaining the unknown by the known, as in certain mathematical proofs. It aims to give an account of what is observed by the properties of what is imagined. They were both geniuses. And this is really at the heart of the matter. And anyway. Roger, thank you for inviting me. I'm sorry, I do apologize for taking on.